Welcome everyone to this webinar, Recommendations of the National Advisory Committee on Immunization, NACI, on the use of Pfizer BioNTech COVID-19 vaccine. My name is Yoav Keinan, and I am an infectious disease clinician and the scientific director of the NCCID, and will be your moderator for today. NCCID is funded by the Public Health Agency of Canada to provide knowledge and to provide evidence for use for public health. Before I begin, I would like to acknowledge that the NCCID is located on the original lands of Anishinaabe, Cree, Oji-Cree, Dakota, and Dene people, and homeland of the Métis Nation. We respect the treaties that were made on these territories and acknowledge the harms and mistakes of the past. We dedicate ourselves to move forward in partnership with Indigenous communities in the spirit of reconciliation and collaboration. There are a few housekeeping notes that I would like to go through before we begin. We are running the webinar through Zoom. If you're having challenges with Zoom, we are also live streaming the event and the presentations on YouTube. You can find a link in the email you received from Eventbrite today, or for new registrants yesterday, or for new registrants today. If you have technical problems with Zoom or accessing the live stream, please email us at nccid@umanitoba.ca. We will do our best to assist you. Today's webinar is to inform healthcare and vaccine providers on the NACI recommendations regarding the use of the Pfizer BioNTech COVID-19 vaccine. The webinar is divided to two modules. The first module, Dr. Mrna will describe the characteristics of the vaccine and summarize the key information, handling, and administration of the Pfizer BioNTech COVID-19 vaccine. The second module, Dr. Robin Harrison will summarize the clinical evidence for the vaccine and the NACI recommendations for the use of Pfizer BioNTech COVID-19 vaccine. Following the two presentations, we will have short Q&A session. Please enter Q&A, your questions in the Q&A tab. We have many questions received prior to this, and we will make attempt to answer as many as we can, but we cannot promise to answer all questions. Lastly, the event is live and being recorded. The recording of presentation and slide will be available on the NCCID website after the webinar. Without further ado, I would like to invite the first speaker, Dr. Warshawski. Dr. Warshawski is a medical advisor at Public Health Agency of Canada working on COVID-19 vaccine rollout response. She is also an adjunct professor at the Department of Epidemiology and Biostatistics at Western and past chair of the National Advisory Committee. She has worked as an associated medical officer of health in Middlesex, London Health Unit and public health physician in Public Health Ontario. Podium is yours, Dr. Warshawski.
Thank you very much, Dr. Keinan. So I have no conflicts of interest to declare. And the objective of this module is to go over the characteristics of the Pfizer BioNTech vaccine and also to summarize some of the key information regarding the handling and administration of this vaccine. So the first part, the characteristics of the vaccine itself. You've likely seen this image before, so you know that the SARS-CoV-2 virus is covered with a spike protein on its surface. And it's that spike protein that binds to a receptor, the ACE2 receptor on our cells, in order to gain entry into our cells so that it can use our cells' machinery to replicate itself and cause infection and illness. So with all the vaccines that we may have available to us in Canada, the goal of those vaccines is to introduce the spike protein to our immune system so that we make an immune response to it. So we either make antibodies and or cellular immune response so that when we see the live virus itself, we already have our pre-prepared protection, which will protect us from infection and hopefully infection and also disease from the virus. So there's two ways, or a number of ways actually, that we can give our body the spike protein in order to make an immune response. We can prepare that spike protein in a manufacturing plant, so we can actually manufacture that spike protein and deliver that as the vaccine so that we make an immune response to that spike protein. Or we can use the cell's own machinery in order to make that spike protein that we then subsequently make the immune response to. So our traditional vaccines would normally make the spike protein in a manufacturing plant and deliver the spike protein directly. But these novel technologies, which are the first vaccines that we will have available, use a technology whereby we provide the genetic code for the spike protein and use the manufacturing capacity within our own cells to make the spike protein and then subsequently make the immune response to the spike protein. So this is how the Pfizer BioNTech vaccine works. It's an mRNA vaccine, which, as mentioned, is a novel technology. None of our pre-existing vaccines use this technology, although there has been lots of study of this technology. This is the first authorized vaccine that we have using this technology. So what happens is that the mRNA, which is the genetic code for the spike protein, is encased in what we call a lipid nanoparticle. So that lipid nanoparticle is basically the carrier that brings the mRNA to our cells. And then the mRNA with the lipid nanoparticle is encapsulated into the cell. So it enters into our cells. And in the cells, the mRNA is released and then uses the machinery within our cells to make the spike protein. And then that spike protein is either released out of the cell or attached to the surface of the cell itself. And it's through that we make both a cellular, so a T cell response, and a humoral, so an antibody response to the spike protein. So basically, we're using our own cells' machinery to make the spike protein, to which we make an immune response. So the mRNA, which is the genetic code for the spike protein, is the main part of the vaccine. But mRNA, we know, is inherently unstable. So in order to get the mRNA from the vaccine in our arm into our cells, we need to incorporate it into a lipid nanoparticle. So the lipid nanoparticle is basically the carrier, a very small carrier that brings up the mRNA from the vaccine into our cells so that we can make an immune response. I kind of think of it as sort of a crystal vase, which is the mRNA that you're trying to get delivered as a holiday present, and you want to make sure it arrives intact, so you incorporate it into some bubble wrap to make sure that it arrives without any of the mRNA disrupted. So that's what the mRNA is, the genetic code, delivering to our cells, wrapped in this lipid nanoparticle to make sure that the mRNA isn't degraded along the route after it's injected, and it actually arrives at our cells, usually our dendritic cells, our antigen presenting cells, and it goes into those cells, uses the cell's machinery to make the spike protein, and then we make the antibody response or other cellular response to that spike protein. So to look at the specific characteristics of the Pfizer BioNTech vaccine, as we've mentioned, it's an mRNA vaccine that we use to translate into the prefusion spike protein to which we mount our immune response. It was authorized for Canada in Canada on December 9th, and it's authorized for 16 years of age and over. It does come with a diluent, and so this diluent is 0.9% sodium chloride or normal saline. And important to use the diluent that's provided by the manufacturer or by the National Operations Center. So don't use any other diluent except that provided with the vaccine or to accompany the vaccine. The dose is 0.3 mils after dilution, and that would be the equivalent of 30 micrograms of mRNA. It is a 2-dose schedule, and it's administered either 21 to 28 days apart from the first dose, and the minimum interval is 19 days. It's intramuscularly administered, and there's no adjuvant in the product. There's also no preservative in the product. It does come as a multi-dose vial where there's enough for at least 5 doses of vaccine in the vial. And it's stored in an ultra-frozen state and will be delivered to you in an ultra-frozen state, so between -80 and -60 degrees Celsius, but it can be stored in the refrigerator for 2 to 8 degrees, and we'll talk a little bit more about this in a minute. All right, so now to delve a little bit more into the handling and administration of the Pfizer BioNTech vaccine. So as just mentioned, it is an ultra-frozen vaccine. So when it comes, it will be in the ultra-frozen state. It can, though, be stored in the refrigerator for at 2 to 8 degrees for 5 days. But if it's in the refrigerator, it can't go back into the freezer, so it shouldn't be refrozen. In the freezer or in the refrigerator, it needs to be protected from light, so you can do that in the original storage container or a similar storage container that you have. The diluent that will be used with the vaccine can be stored at room temperature. So when you receive the product, it will come in a thermal shipper. So this is a special storage container that's filled with dry ice in order to keep the vaccine at that ultra low frozen temperature. So the first thing to do when you get it is to, you can open the box, inspect it, and then stop the data logger. So that's the temperature monitor that will come with the vaccine. It's important to stop that when you get the vaccine, because what that will do is trigger the company to send an email that will indicate that the product has been handled in the appropriate temperatures all through its shipment to your location. And then you have 3 choices for storing the vaccine. One is to put it into an ultra low temperature freezer between -80 and -60. The other is to keep it in the thermal shipper that it came in. So it can be kept in this thermal shipper for up to 30 days. You'll have to replace the dry ice initially, and then at 5-day intervals, you can replace it 5 more times to get to that 30-day maximum storage in the thermal shipper. When you get it, and if you're keeping it in the thermal shipper, you should put a new data logger into the thermal shipper in order to be able to monitor the temperature. And as mentioned, you can also store it in the refrigerator at 2 to 8 for 5 days. You can also take it from the ultra low temperature freezer or the thermal shipper into the refrigerator at any time anytime in the storage times for the ultra low temperature freezer or the thermal shipper. You can move it to the fridge and keep it there for 5 days. However you're storing it, though, when you first get it, you will need to handle the dry ice. So it does require some training and personal protective equipment to handle the dry ice. So eye protection, thermal gloves, and your skin being protected when you're handling the dry ice. If you're not storing it in the thermal shipper, you won't need the dry ice anymore after you've removed the vaccine. So what you want to do there is just let the open up the thermal shipper in an open, well-ventilated space that's secure, so no one can get at the dry ice, and it will sublimate, so it'll eventually just disappear from the box over a number of days. So again, an open, well-ventilated space is where you want to keep it so that the dry ice will just disappear. The transport guidelines have recently just changed. So we do have new transportation guidelines, which do allow some onward transportation of this product in either the frozen state or the thawed state. But if transporting in the thawed state, need to be very careful not to jostle the vaccine around, because again, it's a very fragile vaccine. Okay, so the next step in handling the vaccine would be to thaw it. So if you're stored it in the ultra low temperature freezer, then obviously when you go into that freezer, it's going to be very cold. So you don't want bare hands. You want to wear some gloves or use tongs to get the vaccine out of the freezer. Or if you've stored it in the thermal shipper, again, you need that protection from the dry ice. So the eye protection, thermal gloves, and protecting your skin. So you can thaw it in one of 2 ways, either at room temperature. It will take 30 minutes to thaw at room temperature, and it can stay at room temperature in the thawed state for up to 2 hours. So once you've removed it from the freezer or the thermal shipper, you have 2 hours to mix it with the diluent, but it will take at least 30 minutes for it to be thawed before you can do that. You can also thaw it in the refrigerator. It will take 3 hours about to thaw in the refrigerator if you're thawing a full tray. Just to mention, though, that you don't have to thaw a full tray all at once. In either way, you can thaw just a vial or 2 or whatever you need. You don't need to thaw the full tray. If you're thawing it in the refrigerator, it can stay there, as we mentioned, for 5 days. If you're taking it from the refrigerator in order to mix it with the diluent, you just want to make sure it's very thoroughly thawed. So you might want to leave it at room temperature for a few minutes just to make sure that it's thoroughly thawed. You also want to clearly mark the times that you've done all these movements to thaw. So if you're moving it to room temperature, you want to mark the time that you've done that so that you can check the 30 minutes to make sure it's thawed. And you can also know when it is approaching that 2 hours, because you only have 2 hours to mix it with the diluent. So you want to mark the time you've moved it to room temperature so you know when it's getting towards those time periods. If you're thawing it in the refrigerator, you also want to mark the time that you've done that so that you can know when it's heading towards that 5 days and you need to use it before that 5 days in the refrigerator. Just to note that when it's in the thawed state, it will look white to off-white, and there may be just some amorphous particles. So you can expect to see those. So after you've thawed it, the next thing that needs to be done is to dilute it. So as Robin Lightly mentioned, it needs to be at room temperature for dilution. So that can be either 30 minutes from the refrigerator to room temperature, or it can be, from the freezer to room temperature, or from the refrigerator. It stays for it will take up to 3 hours in the refrigerator. So once it's thawed, then you need to dilute it. You have to dilute it within 2 hours of it being at room temperature with the diluent that's provided. So to do that, you gently invert it 10 times, don't shake it, and then use the alcohol hand rub on your hands. Then you wipe the vial stopper with an alcohol wipe for both the vaccine and the diluent, and allow that to dry. Then you'd want to take a 21 gauge needle or thinner and take 1.8 mils of the sodium chloride diluent out of the vial of the diluent, and then inject that into the vial with the vaccine. With the needle still in the vial, you're going to notice that there's some pressure when you add that 1.8 mils of diluent. So you want to relieve the pressure in that vial by pulling the needle out of the liquid and just keeping it in the air in the vial, and then pulling out the 1.8 mils of air from the vaccine vial. Then you'll take the needle and the syringe and you'll put it directly into the sharps container, and you'll also take the remaining diluent and put that in the sharp container. You only want to use the diluent once. You may have extra volume in the diluent, but you want to throw it out without using it again. And then gently invert 10 times the vial again. This will mix the diluent with the liquid that has been thawed. Again, you don't want to shake this vaccine because it's very fragile with the lipid coating and the mRNA, so you don't want to shake it. But you do want to invert it 10 times back and forth. Then check if there are any particulates or discoloration. You will expect an off-white color. And then mark the vial with the date and the time of dilution, because here you want to be very careful. You only have 6 hours from after you first puncture the vial to use it. That's because of the sterility. There is no preservative in the vial. So you want to make sure that you use it within 6 hours of dilution, and so you mark the date and the time on the vial itself. It can be stored at room temperature during that 6-hour period, and it can be exposed to light in the room, but you don't want it exposed to direct sunlight. If you don't use it in the 6 hours, then you have to discard it, unfortunately, and we want to try really hard not to have any wastage with this vaccine. So you want to have a plan to make sure that you use all the vaccine that you have mixed with the diluent. Next slide, please. So this vaccine it is stable if pre-drawn for 6 hours. And I just do want to talk about pre-drawing a bit here, because although you can do that, it is stable in the syringe. You really want to just pre-draw as little as you can in order to keep your clinic running. So best practice is not to draw up too much in advance, just as much as you need to keep your clinic running, and then have that on hand so that you can keep a steady supply of syringes that are loaded to keep as the clients come into your clinic, but not too much so that you're storing it for too long. So to pre-draw, you use to draw up, you use the alcohol-based hand rub on your hands, swab the vial stopper again, so it with alcohol, and let it dry. One thing to remember is to ensure that before you draw up that the needle is tightly screwed onto the syringe. So really check that the needle is firmly attached to the syringe. And then using aseptic technique and a new needle and syringe, you would draw up the vaccine from the vial. And so we're generally using a 1 ml Lurlock syringe for this. But you can use a 3 ml syringe if the gradations on the syringe are fine enough so that you can actually see each 0.1 ml, because you're trying to draw 0.3 mils. So it's a bit harder when you're using a 3 ml syringe. But if you have a 1 ml syringe, you'll easily see that gradation to get the 0.3 mils drawn up. If you have to remove any air, so if there's any air in the syringe, what we recommend is you keep the needle in the vial itself, so that if you have to express any air, you're not losing any vaccine in that process. And then, of course, you would repeat steps 2, 3, and 4 to get 5 doses from the vial. So here we have had a few questions just to point out that in some situations you can get 6 doses from the vial, and that's okay to use if you can get the full 6th dose from the vial. What we don't want is to be taking a little bit from one vial and another from another vial. So if you can get a full 6th dose from the vial, then that's okay to use. Next slide, please. So finally, it comes to administering the vaccine. So here, alcohol-based hand rub on your hands, of course. We don't recommend the use of gloves for administering vaccines, except if your skin is not intact. But if your skin is not intact, it may not be the best thing to be working in an immunization clinic, because you're going to need to be using a lot of alcohol hand rub, because every time you change your gloves, you're going to need to use the alcohol-based hand rub. So best if your skin is not intact, probably not to be working in an immunization clinic. And we don't recommend the use of gloves for any other purpose except non-intact skin. So here you want to check that the syringe is, there's no particulates, discoloration, and the right dose is in the syringe, and ensure that it's not cold. And then you want to prepare the skin with an alcohol wipe. And this is as per normal, from the center moving outwards, and allow it to dry. And then you're going to give the 0.3 ml dose intramuscularly in the deltoid. And then immediately either activate the safety engineered device if you have a safety engineered device on your needle and syringe, or if you don't, directly discard it into the sharps container. So whatever you do, you don't want to put that needle and syringe down on the workstation, because there's a concern that you may either have a needle stick injury or reuse it accidentally. So as soon as you've used the needle and syringe, discard in the sharps container, and then use the alcohol-based hand rub again. So as we mentioned, the return is 21 to 28 days. The manufacturer has recommended 21 days, but NACI has recommended an interval up to 28 days. Obviously, the sooner the client returns, the faster they'll get their full protection, which would be at 21 days after the first dose. If, however, they come back later than that, if there's a delay, then the recommendation is not to restart the series. Next slide, please. So this just shows you the site for administering of an IM injection. It's the deltoid muscle, so 2 finger breaths below the acromion process, right in the center of that muscle, and that's where you give the injection. I just want to point that there's no recommendation to draw back on the syringe. So when you give the needle into the deltoid muscle, do not draw back on the syringe. There's no need to do that, and it just prolongs the procedure. Next slide, please. So finally, this is just a summary of what has been talked about, the storage at in ultra low temperature freezers at -80 to -60. It can be stored in the refrigerator for 5 days at 2 to 8 degrees Celsius. Here it mentions onward transportation information is forthcoming. We did actually receive some information which we will be disseminating out, that under some circumstances you can do some onward transportation of this product, either in the frozen state, ultra frozen state, or in a liquid thawed state under very confined circumstances, which we will be disseminating that information. As mentioned, it has come with a diluent, and we use 1.8 mils of the diluent. It's the normal saline diluent, and remember to use the diluent that's provided by the National Operations Center and the manufacturer. Important to use it for within the 6 hours after first puncture, because there is no preservative in the product and you don't want to have any issues with contamination. So sterile technique and using it within 6 hours of dilution. The maximum time at room temperature is 2 hours before dilution and then 6 hours after dilution, as mentioned. It is stable if preloaded in syringes, but as we mentioned, you want to just preload enough to keep your clinics running and not more than that. And the dose is 0.3 mils, and the second dose in the 21 to 28 day interval. And NACI has given an interval, a minimum interval of 19 days, but the optimal interval to strive for is around that 21 day mark. But again, it can be as long as 28 days, and don't restart if it's beyond that. And the final slide, I believe. Just to say that there are other webinars that have been pre-recorded and are available on the AMI website, which will complement this webinar. Thank you very much, and again, apologies for the technical problems. Over to you, Raman.
Thank you very much, Dr. Warshawski. The next presenter who has already stepped up in the previous one is Dr. Robin Harrison. Robin Harrison is an adult infectious disease specialist from Edmonton and incoming chair of the NACI Influenza Working Group. She's passionate about policy development to support clinicians and prevent infection transmission. Over to you, Dr. Harrison.
Thank you very much. So I wish to just thank our host today, the National Collaborating Centre for Infectious Disease and the Public Health Agency of Canada. And I'm very pleased to be sharing with you the recommendations from the National Advisory Committee on Immunization on this product. I also do not have anything to declare. I do not have conflicts of interest in relation to this work. So what I aim to do is accomplish these two objectives in the next short while with you. So I hope to summarize clinical evidence for the Pfizer BioNTech COVID-19 vaccine, and then to summarize the National Advisory Committee on Immunization recommendations for use of this product. And as you've just heard from Dr. Warshawski, this vaccine was authorized for use on December 9th in Canada and for individuals 16 years of age and older. So on the heels of that came recommendations from Canada's National Advisory. We abbreviate as NACI. So this is the National Advisory Committee for use of this vaccine. And I really encourage you to look for the full details, the supporting evidence. There's a lot more in the published statement. The web link is on the slide. I obviously won't be able to walk through all of this. I'm only going to hit the highlights, but it is a resource to you, and I encourage you to look at it. I also want to point out that as evidence is evolving, and these are early days, and as we roll through the COVID-19 pandemic, this space you can expect to evolve. This will be evergreen, and so there will be information added as we move through together. I'll just take a brief minute to tell anyone who isn't familiar with NACI what it is. So NACI is 56 years old and an external advisory body to the Public Health Agency of Canada that develops evidence-based advice on vaccines that are approved for use in Canada. And so you know, over the 56 years, NACI has evolved, and in 2020 has been moving through this pandemic rapidly with you and everybody globally. And so in order to provide these recommendations, NACI is comprised of experts in the fields of pediatrics, infectious diseases, immunology, pharmacy, nursing, epidemiology, pharmacoeconomics, social science, and public health. So this interdisciplinary group, voting members, and also others supporting this work is what lead to the recommendations, and this is going to be one example. NACI shares this information with the public in the form of NACI statements, and those statements are then synthesized into our Canadian Immunization Guide, which many of the frontline practitioners on the call will be very familiar with. So our first objective, to review the clinical evidence for the Pfizer BioNTech COVID-19 vaccine. This vaccine has been studied through clinical trials. They are the trials that you would expect for study of a new vaccine product. We call those phase 1, 2, 3. For safety first and foremost is what these vaccines will start with in phase 1. Then what you've seen through the pandemic, you've noticed, I'm sure, that things are moving quickly. And so you'll see the phase 2 and 3 trials, for example, being done in concert. And that brings us to present day, where there's been an interim analysis of the evidence for this particular product. So from these phase 1, 2, 3 trials, we can see there is some evidence available already on immunogenicity. This is looking at the body's immune response to the vaccine measured through tests, blood tests. And this is available for adults in the 18 to 55-year-old range, but also I want to highlight in the 65 to 85 plus range. And this is significant when you consider the pathogen. This pandemic is not like the last one in that although elderly individuals are vulnerable every year to seasonal influenza, and the last pandemic, which was an influenza pandemic in 2009, this COVID-19 pathogen has a very clear risk for those who are older in terms of severe disease and death. And so it's important to note that older individuals have been included in some of these trials for vaccines generally. There's also evidence on safety. As I mentioned, it starts in phase 1, continues throughout, and even post marketing of vaccines. And then finally, efficacy, which in a trial that is, does this vaccine work? And we speak about that as efficacy in the context of a trial. So the phase 2/3 portion of the trial for the Pfizer BioNTech product licensed in Canada was involving approximately almost 44,000 study participants, and they were randomized 1-to-1. You either received a vaccine or a placebo if you're a participant entering this trial. The trial data that we have to date are for an interim analysis of that data. And the time of follow-up varies for the individuals in the trial because it's not done, the study is ongoing, but what we know is out to 4 months, 14 weeks is the maximum. So had any one of you been looking to enroll in this trial, you would have been excluded if you were known to be pregnant or breastfeeding, immune compromised, had you had a known clinical diagnosis of COVID-19 based on your clinical illness PCR test, or if you were known to have an adverse reaction associated with the vaccine. Also, those who had received blood and plasma products or immune globulin would be excluded. It's important to note that people included in the trial had a myriad of chronic conditions. So there were individuals with medical conditions, including some that would put them at risk for COVID-19 that would have been included in the study. And people who had had a blood test to look for serology or evidence of a possible past SARS-CoV-2 infection were not excluded. So there were some of those individuals, and there were also some individuals from long-term care facilities, although the numbers are limited. So the question that everyone was waiting for is what did the study show and what was found? So the answer here, I think many of you know, this vaccine was efficacious. So their primary endpoint here is against symptomatic confirmed COVID-19 disease. So when you look at those who received placebo versus those who received vaccine, it appeared the vaccine is 95% efficacious in preventing symptomatic COVID-19 disease. The highest efficacy was seen after the second dose. It is a 2-dose series, and the peak humoral or antibody response, and also the cellular immune response, T cells response, which are important for viruses, occurred after the second dose. There is currently insufficient evidence on how long that protection lasts. As I've just mentioned, these are early days, and this study, it will be ongoing. And so we do expect information to come, but we still won't be able to know definitively how long protection will last. It wasn't, we can't comment yet on how it specifically prevents death, hospitalization, or infections without symptoms, or transmission in between people. But these are things that will be coming. The studies are ongoing. Safety. So of these 37,700 trial participants that completed a median follow-up time of 2 months after the second dose, and about half of those, as we said, in placebo and the other half in the vaccine arm, there were no serious safety concerns identified to date, but the studies are ongoing, and this is routine, and so this makes no exception. when there is an adverse event, they are categorized and as being very common, rare, and so on. So the very common adverse events were described. And so by definition, that means affecting more than 10% of people. They tended to be mild or moderate and transient, resolving within a few days. And to give you an idea of what these types of symptoms were, it was pain at the injection site, fatigue, headache, muscle pain, chills, joint pain, fever. And some of these adverse events appeared to be more frequent after the second dose. Now, there's currently minimal evidence to inform on differences in safety and efficacy between those who did and didn't have evidence of SARS-CoV. Remember, there were people that had serology, blood tests to suggest they'd had a past SARS-CoV infection, but there is some minimal evidence on that topic. So now I'm going to take us to our second objective. These are the NACI recommendations for use of this vaccine. Whoops. Sorry about that. Number one, NACI recommends that a complete vaccine series of COVID-19 vaccine should be offered to individuals in the authorized age group who don't have contraindications to the vaccine. Now, as we start rollout in the context of limited vaccine supply, initial doses should be prioritized for key populations that are outlined in the NACI guidance on prioritization of initial doses. And this is a strong NACI recommendation. This reflects the evidence base that we have to date. And so this is the guidance. There, you'll see in the statement, there is rationale below each of the recommendations, and they're not all presented here on the slides, but there are a few comments here for you to see. And that this recommendation obviously hinges on the good evidence of high efficacy, and we can comment on the short term and look forward to being able to comment on longer term as time goes on. The second recommendation, NACI recommends that all individuals should continue to practice recommended public health measures for prevention and control of SARS-CoV-2 infection and transmission, regardless of vaccination with COVID-19 vaccine at this time. This is also a strong recommendation, and this reflects the items I just mentioned in that we don't yet know the answer to how long is this going to last, and we also haven't yet had time to explore whether this prevents asymptomatic infection and transmission of virus from one person to another, even in the vaccinated. What's been looked at is the vaccine protecting the individual who is immunized. So at present, this is the recommendation. The third recommendation I want to share is those with previous infections. So NACI does recommend that a complete series of vaccine may be offered to individuals in this authorized age group who don't have contraindications, who have had previously PCR confirmed COVID-19. So in the context of limited vaccine supply, initial doses may be prioritized in provinces and territories to individuals who've not previously had SARS-CoV-2 infection, but it can be given to those who have. And so that's the key thing I want to impart here. Now, in the rationale, you'll see that there is a lack of evidence in this group. And so there are still some unknowns, but it is felt that there's no safety concern with this approach in all the information we have to date. And so it's recommended this can be done. There are some suggestions you'll see in the rationale that you may delay or may choose to delay for 3 months, again, looking at short supplies, because reinfections, although they've been reported in people who've had a COVID-19 infection, they have been rare within the first 3 months. I'm moving now on to subpopulations. So this is a recommendation for immunocompromised or immunosuppressed individuals. NACI recommends that the vaccine should not be offered routinely to populations who are immunosuppressed due to disease or treatment, or those with autoimmune disorder until further evidence is available. However, a complete series may be offered to individuals in this age group if a risk assessment factoring in their risks of exposure and disease or severe disease outweigh the potential risks for that individual, and if there is conformed consent about the fact that we don't have data on this population because they were excluded from the trial. I would just want to point out that, as I mentioned, people with medical conditions were enrolled in the trial, and that did include people who are living with HIV, and those that have well-controlled HIV are not considered immune compromised, and so this would not include those individuals. The same recommendation applies for pregnant and breastfeeding individuals. So it should not be routinely offered to people who are pregnant or breastfeeding and could be deferred until after the pregnancy. However, it may be offered to pregnant individuals in the authorized age group if a risk assessment deems that benefits outweigh the potential risks and there is consent, sort of discussion and informed consent. And this is a discretionary recommendation. And I'm going to just take you through one more special population here. This is children and adolescents. So you'll note that it's authorized for use in those 16 years and older, and it should not be routinely offered to those in a younger age group. However, it may be offered to individuals 12 to 15 who are at very high risk of severe outcomes of COVID-19 and at increased risk of exposure if there's informed consent with a parent or guardian and discussion about the lack of evidence in this population. And as I've said before, there are data coming and trials going, including addressing this age group. Okay, so now I just want to speak to the contraindications, and I don't have too many more slides, and I'm cognizant of the clock here, so I'll just mention that this would be contraindicated in anyone who had an anaphylactic reaction, say for example to the first dose of the vaccine. The individuals with a history of severe allergic reaction to a component of the vaccine should not receive the vaccine. The component of interest is polyethylene glycol, a potential allergen in the vaccine, and it is that allergen is known to cause type 1 hypersensitivity reactions. And so that would be a contraindication to this vaccine. If there is a specific concern about a possible allergy to a component, then an extended period of observation would be recommended. And so for us in our immunization clinics in Canada, that would be a 30-minute as opposed to 15, which would be routine. So there are a few precautions. The first one relates to bleeding disorder. So individuals who are taking anticoagulation can safely receive this vaccination. Individuals with bleeding disorders should ensure that that's been optimally managed prior to the intramuscular injection, the concern of bleeding. The second one is people with acute illnesses should not have this vaccine at the time of that illness. It would be prudent to wait until the symptoms have resolved. The fourth thing I want to highlight is that, as we've mentioned, vaccination of individuals who may be currently infected with the virus, it's not known to have a detrimental effect on the illness. However, the vaccination should be deferred for several reasons. You don't want to have any confusion about is an individual having a vaccine side effect or complication of their infection and illness, and also to prevent transmission of infection to others. Those who are ill should be isolating, and so not in an immunization clinic until their illness has resolved and they've completed 10 days of isolation for the communicability of this pathogen. Obviously, if someone arrives with symptoms, they would be redirected and you'd have a public health guidance on that topic. You've heard from my colleague, it's about the schedule. And I'm just going to point out that at present, this is the only product we have approved for use in Canada. In the event that there were other products, the same product ideally would be used to complete this vaccine series. You don't need to do antibody testing before or after receipt of this vaccine. So you wouldn't need to test antibody response before or after. It's not necessary, not additive. And as you saw, the efficacy in clinical trials is high with this vaccine. It also should not be given simultaneously with monoclonal antibodies or convalescent plasma, and there is some guidance on that topic in the statement. Prophylactic medications to prevent fever or side effects is not recommended. However, for those individuals that have a sore arm or fever or symptoms after their vaccination, these can safely be taken, and so examples would be acetaminophen or ibuprofen, and again, there's guidance in the statement. And so with that, I'd like to just close. I'm sharing with you here the NACI membership, the liaison representatives, and the ex officio representatives. So you can see the breadth, the number of individuals that contribute to the work of NACI. I would like to make particular mention of the NACI Secretariat, who has done an incredible work this year. And this is the High Consequence Infectious Disease Working Group working on guidelines like this. You see the chair, the vice chair, and the leads from the Public Health Agency of Canada and the Secretariat. There are 3 key publications that this builds upon, and if you haven't had a chance to look at them, I really encourage you to do so. One are the research priorities. This was the first step, setting those research priorities so that we could inform policy in the way that you see unfolding now, and preliminary guidance on key populations for early immunization, and then guidance on prioritization of these initial doses. These are the documents that led up to the current statement, Evergreen, and that I refer you to. And I'm going to close there. Thank you.
Thank you very much, Dr. Harrison. We are now going to begin our Q&A period with our speakers. So I will go through several questions of the many questions that have been tabled, and I welcome both speakers to address them as they see fit. The first question is, can mRNA vaccine damage or change DNA? Has that been studied?
Do you want me to try to? I can start with that one. So the mRNA does not interact with our DNA at all. So in order for it to work, it has to gain entry into the cytoplasm of our cell. So it never goes near the nucleus of our cell where the DNA of our cell is housed. So it only works within the cytoplasm and never near the nucleus. So there's no possibility of interaction with our DNA at all. So that's not at all a concern with these mRNA vaccines.
To complete that question in terms of risks of sterility, there was a question about that. And the second question is, how was the vaccine made so quickly?
Sorry, was the question with the sterility of the vaccine?
Yeah, the sterility caused by the vaccine. That was a question. So it's related to your previous answer. So just to enforce that.
Yeah, so there would be no interaction with sterility or anything to do with reproduction at all with the vaccine. It has nothing to do with our own DNA. It doesn't interact with it, interfere with it, get anywhere near it. It's totally separate from our own DNA, so there's no chance of that at all. In terms of how the vaccine was made so fast, it is actually because of the mRNA technology that it was made so fast. It's really, you don't have to manufacture the protein in a manufacturing plant, which takes lots of time and lots of technological infrastructure to do. You really just have to get the genetic sequence, which was very quickly available after the virus became known. So we knew the genetic sequence, and right after that, the manufacturers could begin using the technology that they had been developing for quite a long time for this kind of emergency, so that they could just insert the genetic sequence and then manufacture that, and that genetic sequence is what we are given in the vaccine, so that we make the protein for the spike protein and mount an immune response for that. So it's really this technology itself that allowed it to be made so quickly.
Thank you very much. Can you answer what is the lifespan of the viral mRNA in human cells, and how long do we know if it continues to encode and produce spike proteins?
Good question. I don't think we know for sure. We know mRNA is fragile and it does not last very long. So it's in the order of days that it will, circulate around and then eventually be destroyed. But I don't think we know exactly how long it persists for. I don't know, Robin, if you have any additional information that you want to add there.
Yeah, probably not. I mean, you can hear, as you said, in the order of days. The precise number of days, I don't think we know, but days and not longer. I mean, that's part of the reason. It's a labile thing. That's part of the reason of the complicated delivery model. Thank
you very much. Why are there differences in terms of stability between the two mRNA vaccines, and what makes the Mrna vaccine, once it becomes available, different in terms of storage temperatures and stability?
Yeah, so the mRNA sequence for the two vaccines is, as I understand, identical. So it's not the mRNA that would change the stability. It's the lipid nanoparticles that they use are slightly different between the two vaccines, and therefore the stability requirements are different between the two. As mentioned, the Pfizer BioNTech vaccine needs to be stored at ultra low frozen temperatures, -80 to -60, whereas the Mrna vaccine, which is very similar in its technology, has to be stored at -20. So it has to do with the lipid nanoparticle and the way that it's manufactured, and you should definitely not use, mix the two up. So the Pfizer product is the ultra frozen and the Mrna product is frozen in regular freezer temperatures at -20.
Thank you very much. Is there a risk of protein deposition in tissues inducing any maladaptive responses, such as an allergy to prion disease or inducing autoimmunity?
Well, I can comment. I think a key thing to keep in mind here is that, so is there any evidence of that? No. So I think the key thing to keep in mind is that it is early days in terms of the follow-up, as we've highlighted, but when you think about what's happening, you've got this transient mRNA. It's going to make a protein that's going to be a transient thing. It's going to trigger the immune response in the way that our bodies are designed to do, to identify what looks like the spike protein for the pathogen. So it's achieving that, and it's not that it's creating other proteins to cause harm, and these are not changes that are long lasting. As you just heard, this is not affecting your DNA. And so all of the study that looks at the immune response and that looks at the safety of this over time, and at present, you know, you have this in tens of thousands of people, will be looking for any concerns, because you mentioned autoimmune, but no, there's been no concern in that regard to date. And as I said, it started with phase 1 safety, progressing through phase 3.
Thank you very much.
Yeah. I don't know if you have anything to add, Dr. Warshawski, but.
No, that was good. Thank you.
We know that elderly have some degree of immune compromise, and that immune function is better at a younger age. How does this concept apply to this vaccine, and how do we explain the fact that it seems to protect the elderly as well?
Well, so that's a really good question. I mean, one thing that is really good to see here is that older individuals were included in the trials right from the beginning. So this is really important, and this is not always the case. As you mentioned, when you're trying to show benefit, you might choose to enroll those where you think their immune system will be at its prime peak performance, and there is a concept of immunosenescence, and that our immune systems may not work as well as we age or work differently. And so I think it's very valuable that older individuals are in these trials, and it's very encouraging that they too are showing evidence of an immune response. That's just, quite frankly, just a very helpful thing. This is new to us, using a vaccine in this way. Now, we use protein triggers to invoke the immune system, but this is a bit of a unique design where you provide the mRNA. And so I can't answer the why, except to say that it's really important that it's being looked at, and the results are very encouraging, which is a good thing, especially considering the pathogen. You know, this is where the risk is heightened.
Thank you very much. And another group where the risk is heightened is immunocompromised and individuals with cancer receiving chemotherapy, where the risks may be high for morbidity and severe disease in COVID. Would you advocate for immunization versus waiting with immunization, as this group is currently not recommended to be immunized?
Yeah, so at this early stage, what you see in the NACI guidelines are reflection of the evidence. So we're telling you through those recommendations what we know. And so we've spoken about the populations that were enrolled in the trials and where the data lie. But you see there also in the discretionary recommendation acknowledgement of this fact that each individual will have risks, that some may have heightened risk of exposure based on where they live, where they work. There are factors that increase the risk of exposure to the pathogen, and then risks of severe disease. And it's really important to remember when you mention immune compromise, it's a very heterogeneous group. So their underlying disease may lead to the immune compromise, the medications may be, some are very immune compromised. It's a whole spectrum, as the experts in this field know all too well. So I think the recommendation allows for an individual risk assessment for that person, factoring in their risk of exposure, their risk of severe disease, and then a conversation with informed consent that there are not data at this early stage, but they will be coming. And so at this time, what you see that recommendation that says it's not routinely recommended, but for those identified as exceptionally high risk and where you're able to have an informed conversation with their practitioner, that it can be considered.
Thank you. As the virus mutates, do we know about the immunity that the vaccine affords against mutated spike proteins and mutated viruses that we hear in the news that are circulating?
Sorry, the question was, do we know about those mutations and are they affecting the spike protein? Is that the question?
Are they affecting vaccine protection and efficacy?
Right. So this is a space that will be actively, it is being actively monitored. So thus far, those mutations do not appear to affect the spike protein, and so that it wouldn't interfere with, for example, the efficacy of this vaccine. But it is, as I say, being monitored. Viruses, we know they mutate. Now, they don't all mutate at the same rate. Some are worse than others. But as you mentioned, there have been some mutations identified, but so far not interfering with this area, which is good so far. But Dr. Warshawski, do you have anything else to add to that?
No, just to say that, I mean, obviously the vaccine was studied with the range of viruses and their genetic components that were circulating at the time and demonstrated that 95% efficacy, which is very reassuring. But obviously, as you mentioned, Dr. Harrison, we're going to be monitoring this very, very carefully and watching the vaccine effectiveness over time.
Thank you for that. Does the vaccine effectively reduce hospitalizations and deaths? And does it prevent, do we know anything about its ability to prevent transmission or shorten the carriage of the virus?
So the trials didn't specifically look, and the results we have in this interim analysis don't specifically address hospitalization and death, but there are some data on severe disease. And so if you look at what's happened, you had the group that had placebo and the group that had vaccine. And the group that had vaccine, there are very few cases. So there were 170 cases of COVID-19 disease, and 160, two of those in the placebo arm. And so if you then look at those individuals in the placebo arm and the vaccine who actually got disease, you saw a similar trend where those in the placebo group had severe disease more often when you compared to those who had vaccine. So that does suggest there's efficacy against severe disease, estimated to be about 90%. And so for me as a clinician, it follows that if it's preventing severe disease, it would be likely to prevent ICU and death. But to be clear, that's not the precise endpoint to date, but this is coming. You asked about transmission as well. That is also coming, and it is typical at this stage to first look at the direct protection of the individual, and then the next question of what about transmission to others. But with that in mind, it does inform policy about what we know so far.
Thank you very much. And another question is the vaccine protection. Do we have any indication from all stages of the clinical trials of the vaccines for what we can expect in terms of durations and of protection, and will we be needing a booster? I know that some of the data is still not there, but what's your guesstimate?
well, so being very clear, just a guesstimate, but it's not as though you're seeing graphs that look like they're falling down already. So we only have, you know, a median of 2 months here, but things look robust, which is very good. Immune response looks robust and brisk. You know, 7 days after that second dose, you see these. It's where you get your peak signs of response, and it's looking very good. How long it's going to last, we can speculate. I guess I probably, you know, shouldn't do too much speculation, but having both an antibody response and a cellular response is a good thing. But those data have, you know, will be coming, and this is interim, and what we really have right now are the clinical disease efficacy data. And so far so good is what I would say there. But I couldn't comment on whether you would or wouldn't eventually need a booster. That will take time.
Thank you very much. The last question we have time to answer today is related to allergic reactions. Do we know who is most at risk, and what are some of the allergic reactions that have been observed in the clinical trial leading to registration?
Do you want me to?
Do you want to go ahead, Robin?
Sure. Well, I think so. The allergists and immunologists are looking at this, and the polyethylene glycol, as mentioned, is the substance of interest. Now, it can be found in some other products, and so that is actually outlined. And you know, you'll see detail in that NACI statement on the other types of things that could be relevant to that. There's a few things to note in the list there. You know, it's a substance we use in bowel preparations, for example, for colonoscopy. There's some interactions with some food allergies, contact lens solutions. You know, there's a few specific things, people who are known to have allergies to those things. And this will, as I said, it is articulated in the statement, and but they are precise. It's not all comers, so it's not everyone who has an allergy to you know, anything. It's fairly precise. And as I say, allergists and immunologists are going to help to make sure the list is easy to work with, like clear and precise.
Thank you very much, Dr. Warshawski and Dr. Harrison, for excellent presentations and for your sticking around, answering all the questions. I know there are many questions that we couldn't get to because of the volume of participants. I acknowledge the colleagues at the Public Health Agency for their collaboration on the webinar, and our team at NCCID for putting this together. Thank you for joining us for this webinar. We hope that the information that was provided will help you in your practice during these unprecedented times of COVID-19. NCCID will be hosting more COVID-19 vaccine webinars. Please check our website at nccid.ca for more information and promotion on our social media. Before I conclude, I would like to ask you to fill short evaluation when you leave the webinar. The surveys are really important for us for future planning and for improving the webinars that we deliver. Thank you very much.